Okay, so welcome back to this next video in which we are discussing asthma and anti-asthmatic drugs. Okay, so we're currently in the process of discussing how mast cells can be activated, and we're specifically looking at how mast cells can be activated by the allergen binding to IgE molecules that are loaded onto Fc-epsilon-R1 receptors on the surface of the mast cell. Okay, and remember that this is only relevant for atopic asthma where you do actually produce IgE molecules against the allergen. Okay, but most asthma cases are atopic asthma cases. Okay, so we've discussed that what will happen is when the allergen arrives in the extracellular fluid surrounding the mast cell, you're going to get the cross-linking of these Fc-epsilon-R1 receptors together, and this is because they'll have different IgE molecules bound to them which can bind to different epitopes of the allergen. Okay, these cross-linked Fc-epsilon-R1 receptors then translocate into these special portions of plasma membrane known as lipid rafts, where you have the linker for the activation of T cells protein present. Okay, and through mechanisms that are not fully understood, the Fc-epsilon-R1 receptors that are cross-linked together activate the Lin and Syk free tyrosine kinases, which phosphorylate tyrosine residues on the intracellular aspect of the linker for the activation of T cells proteins. Okay, this then results in there being loads of these phosphotyrosine residues for the docking of proteins, and we've discussed that one of the pathways that this can then activate results because phospholipase C gamma enzymes, which have SH2 domains and are therefore capable of binding to phosphotyrosine residues, will be recruited up to the underside of the plasma membrane by binding to phosphotyrosine residues on the intracellular aspect of the linker for the activation of T cells, and then they themselves get phosphorylated and that activates them. Okay, and then they're both nearby their substrate and now activated, so they're going to break down PIP2 molecules within the phospholipid bilayer to diacylglycerol and IP3, and the IP3 will then drop into the cytoplasm and cause the release of calcium from the endoplasmic reticulum through IP3 receptors, and that calcium will then cause the degranulation of the histamine-containing granules, and histamine is one of the key molecules that then goes off and tells bronchial smooth muscle cells to contract. Okay, now I want to discuss another pathway which is activated by these phosphotyrosine residues on the intracellular aspect of the LAP protein, and this pathway is going to result in the production of cysteine leukotrienes. Okay, right. So ignore the fact that those two lines have suddenly appeared there. They're just going to be helpful for us now. So basically, we'll draw this picture out again. This is our lipid raft-like membrane. Okay, we now have our linker for the activation of T cells in this lipid raft-like portion of the plasma membrane. Okay, and we'll colour this in turquoise again. Okay, and we know that this linker for the activation of T cells has now got many tyrosine residues on its intracellular side which have been phosphorylated. Okay, so I'll show these here. So here are the tyrosines on the intracellular side of the linker for the activation of T cells, and they have now been phosphorylated, so they're now phosphotyrosine residues. Okay, now, what then other targets beyond phospholipase C gamma enzymes can be recruited to these phosphotyrosine residues? Well, the other target that I want to talk about is something called the growth factor receptor bound protein 2, okay, which is capable of activating the Ras-Raf-MEK-ERK pathway. Okay, so let's see this. So basically, let's start with growth factor receptor bound protein 2 then. Okay, now this protein again has one of these special domains that is capable of binding to phosphotyrosine residues, and this is one of these SH2 domains here. Okay, and this is how the protein can bind to these phosphotyrosine residues on the intracellular aspect of our linker for the activation of T cells protein. Okay, so this is this protein, Grb2 for short, and this stands for growth factor. That's the G, and then it's growth factor receptor bound protein 2. So it often is recruited to phosphotyrosine residues on the intracellular aspect of a growth factor receptor, which are examples of receptor tyrosine kinases usually. Okay, so growth factor receptor bound protein 2. So the G stands for growth factor, the R stands for receptor, the B stands for bound, and then it's protein 2. Now, growth factor receptor bound protein 2 then has this SH2 domain which is capable of binding to these phosphotyrosine residues, but it also has another important domain, okay, which I'm going to have here. And this other important domain is what's known as a Sark homology-free domain, which I'm now colouring in orange here. Okay, and for short, the Sark homology-free domain is abbreviated to SH3. So this is Sark homology-free domain. Okay, and Sark homology-free domains bind to portions of proteins that are proline-rich. Okay, so these bind to proline-rich portions of proteins. So SH2 domains bind to phosphotyrosine residues, SH3 domains bind to proline-rich portions of proteins. Okay, so we've got the Grb2 protein then now recruited to the underside of the phospholipid bilayer by it binding to the SH2 domain. What can now happen once it's bound to that phosphotyrosine residue of the LAP protein is that another protein can now bind to the SH3 domain of Grb2 here. Okay, so I want to stress that this green, white and orange protein, which looks like some flag that I don't know which country it actually is for, this is one whole protein basically. It's not 3 separate proteins. This is just different domains of this protein. Okay, and now what can happen is the SH3 domain can bind to another protein called SOS. Okay, so I'll just colour in SOS in red here. Okay, now, once SOS then has been recruited to the Grb2, what it's now going to do is it's going to act as a Ras-Raf-MEK-ERK. Okay, so SOS is what's known as a Ras-Raf-MEK-ERK. So what does this stand for? Well, Ras is a type of monomeric G protein. Okay, so let's just discuss Ras monomeric G proteins. So Ras monomeric G proteins, like all monomeric G proteins, can be in 2 states. Okay, they can be in an off state where they have guanosine diphosphate bound to them, and they can be in an on state where they have guanosine triphosphate bound to them. Okay, so monomeric G proteins, these are individual proteins. Contrast that to heterotrimeric G proteins, which we'll see later on in this video, okay, where you have 3 separate subunits. These are just a single protein, okay, and they can be in 2 states. They can be in the off state, in which they have guanosine diphosphate, GDP, bound to them, and they can be in an on state, where they have guanosine triphosphate, GTP, bound to them. Now initially the Ras protein is going to be in the off state, and it's going to have guanosine diphosphate bound to it. So here is our Ras monomeric G protein. Okay, so I'll colour in the Ras monomeric G protein in red. Now I should say that there are multiple different types of Ras monomeric G proteins. Okay, there is what's known as NRAS, HRAS, KRAS. Okay, so these are all different types of Ras monomeric G proteins. However, for our purposes, they all do exactly the same thing. So we're just going to abbreviate them all down to Ras monomeric G proteins. Okay, now, they are not an integral membrane protein. However, they are attached to the underside of the phospholipid bilayer, which is why I've shown the phospholipid bilayer just above this protein here. Okay, and the way that they are attached is by a lipid moiety, which is this orange line here. So basically the idea here is that you attach on some molecule which is very hydrophobic, which sticks off the side of the protein. Okay, and being very hydrophobic, it's not very water soluble. Okay, so that is going to end up implanting into the inner leaflet of the phospholipid bilayer and then holding the Ras monomeric G protein attached to the underside of the phospholipid bilayer. So Ras monomeric G proteins are attached to the cytoplasmic face of the plasma membrane, basically. Okay, right. So coming back to SOS then, SOS is what's known as a Ras-Gef. Okay, and what does GEF stand for? We now understand what Ras is. Okay, GEF stands for guanine nucleotide exchange factor. Okay, and basically what this means is that it is going to activate the Ras monomeric G proteins. Okay, Ras is currently in the off state. It's currently not doing anything because it's got the GDP bound. Ras-Gefs, Ras guanine nucleotide exchange factors, are going to bind to the Ras monomeric G proteins and catalyze the exchange of the guanine nucleotide here, catalyze the exchange of guanosine diphosphate for a guanosine triphosphate molecule from the cytoplasm. Okay, and they're therefore going to turn the Ras monomeric G proteins from the off state, in which they have GDP bound and are not doing anything, to the on state where they have GTP bound and they can do a lot once they're in the on state. Okay, so that's what is meant by a Ras-Gef. It's something that's capable of activating Ras monomeric G proteins. Okay, so when SOS binds to Grb2 then, like so, it's going to become activated, and now it's near the plasma membrane where the Ras monomeric G proteins, which are in the off state, are. Okay, so what can happen is Ras monomeric G proteins in the off state with guanosine diphosphate bound to them can bind to the SOS protein, and then that catalyzes the release of the guanosine diphosphate from the Ras. Okay, so if I just show this here, here is our Ras monomeric G protein now binding to our SOS protein. Okay, so here is Ras here. Okay, and what this is now going to cause is it's going to cause the release of the guanosine diphosphate from the Ras, and now a guanosine triphosphate molecule is going to come in from the cytoplasm and replace that GTP molecule. Now once that GTP molecule has come in and bound to the Ras changes conformation. It breaks away from the SOS protein now. Okay, it still remains attached to the underside of the plasma membrane, so if I continue the plasma membrane on here, it's still going to be attached to that. Okay, but it goes off and cleaves away from the SOS monomeric G, sorry, not the SOS Ras-Gef. Okay, SOS is not a monomeric G protein, it's the GEF that activates the Ras monomeric G proteins. So it will cleave away from the SOS protein, and then the Ras in the on state with guanosine triphosphate bound to it is then capable of having all sorts of signaling effects. So let's turn over the page and see what the on state Ras monomeric G proteins are going to do. So here again, this is the plasma membrane. Okay, and then attached to the underside of the plasma membrane here, we have our Ras monomeric G protein here, which is now in the on state. It now has guanosine triphosphate bound there, and I'll just colour in the Ras monomeric G protein in red here. Okay, and now this Ras monomeric G protein in the on state is going to now activate other things. Okay, now what does it activate then? Well, basically it binds to a protein that is usually in the cytoplasm, okay, which I'll draw here, which is a serine threonine kinase enzyme. Okay, so this is a serine threonine kinase enzyme, and it's by the name of RAF. Now, just like there is not just one Ras enzyme, one Ras protein, there is not just one RAF enzyme. Okay, there are actually loads of RAF enzymes, but for our purposes they all do the same thing, and therefore we will just abbreviate this down to RAF. So a Ras monomeric G protein binds to a RAF enzyme and recruits it to the plasma membrane like so, forming a complex with it. Okay, and this activates that RAF kinase enzyme. Okay, so the RAF wasn't previously active. When it was just floating around in the cytoplasm, it wasn't active. Now it's bound to our Ras monomeric G protein in the on state. Now it's active. What's it going to do once it's active? Well, it's going to phosphorylate serine and threonine residues within proteins, basically. And one of the key proteins that it now phosphorylates is another serine threonine kinase enzyme, but which is slightly more than just a serine threonine kinase enzyme. Okay, this next enzyme is an enzyme called MEK. okay? And this is more than just a serine-threonine kinase. It's a serine-threonine and tyrosine kinase. Okay, so it's capable of phosphorylating serine, threonine, and tyrosine residues within proteins. Now again, just as there was not just one Ras, there was not just one Raf, there is not just one MEK enzyme, but we're keeping this general. Okay, so we will just say a Ras binds to a Raf, activates that Raf, and then the Raf is going to phosphorylate serine and threonine residues within the MEK enzyme. Okay, so here we go. There is the phosphate group being added onto the MEK enzyme. And I do not know exactly how many serine or threonines get phosphorylated in the MEK by the Raf. Okay, but this conveys the basic principle that the Raf is phosphorylating the MEK enzyme. Okay, it might not be identically one, it probably isn't just one, okay, but it gets across the principle that this is phosphorylating the MEK enzyme. Okay, so what does the MEK enzyme stand for? Well, its name stands for MAP kinase. Okay, that's what the M is for. And then it's /ERK. Okay, that's what the E is for. And then it's the kinase enzyme for MAP kinase or the ERK enzyme. So basically, this is capable of phosphorylating another enzyme which has two names, MAP kinase, and its other name is ERK. So let's see this next kinase along here. Okay, so this next kinase has these two different names. You can either call it the ERK enzyme, or I think slightly more commonly now than the ERK enzyme, it's called MAP kinase, M-A-P-K for short. Okay, so here is the MAP kinase enzyme in orange here, but you might also see the MAP kinase enzyme called the ERK enzyme. Okay, so be aware of that. Now, the MEK enzyme then, which is a serine, threonine, and tyrosine kinase, is going to phosphorylate many residues in the MAP kinase, and I know this time that it's certainly more than one that gets phosphorylated, okay, because it phosphorylates some serine and threonine residues and also phosphorylates some tyrosine residues in the MAP kinase, so it's essential that it has both of these catalytic activities here. Okay, right. So, what does MAP kinase actually stand for? What does the MAP stand for? Well, it stands for mitogen activated protein kinase. Okay, so the MAP stands for mitogen activated protein. So that's what MAP stands for, and then the K is just for kinase. Right, what does ERK stand for? So if you want to use the other name for this enzyme, which is ERK, ERK stands for extracellular signal. Okay, that's the E. Okay, and then it's extracellular signal regulated kinase. Okay, because often this enzyme is activated by signals that come from the extracellular fluid. So ERK stands for extracellular signal regulated kinase. So those are the two different names that you can use for MAP kinase, or you can call it ERK. Okay, right. Now, you should also be aware that the Raf and the MEK enzymes also have other names. Okay, so the MEK enzyme can also be called the MAP kinase enzyme. So you might hear it referred to as MAPKK, okay, because it is the kinase enzyme for MAP kinase. Okay, so you can also hear MEK called, and I won't underline it in orange, I'll underline it in red. You can also hear MEK called MAP kinase, and even worse than that, you can hear Raf enzymes called the MAP kinase, because it's the kinase enzyme for the MAP kinase enzyme, which is then the kinase enzyme for the MAP kinase enzyme. Okay, so be aware that there are these other silly names for these enzymes as well. Okay, and because people don't generally like using this name because it reminds us of a horrible portion of history, you often see that abbreviated down to MAP3K, like so, or you can hear MAPKK abbreviated down to MAP2K, like so. Okay, right. So this cascade then is called the Ras-Raf-MEK-ERK pathway. So you'll hear this cascade that we've just been through called the Ras, that's the first component, then it's the Raf, then it's MEK, and then it's ERK, which is that other name for MAP kinase. Okay, so basically the Fc-epsilon-R1 receptor can also activate this Ras-Raf-MEK-ERK pathway, and it's the activation of the MAP kinase or ERK enzyme at the end here that is going to lead to the production of cysteine leukotrienes. Okay, so let's now study what does MAP kinase actually do once it's been activated. Well, it is now going to phosphorylate and activate an enzyme called Phospholipase A2. Okay, so let me show this enzyme here. Okay, so we'll have phospholipase A2 here, which is another phospholipase enzyme, but it's going to break down very different phospholipids to the ones that phospholipase C enzymes broke down. So this is PLA2, and I'll just bring this up a little bit. Okay, which is phospholipase, that's the PL, and it's phospholipase A2. Okay, right, so let's just colour in phospholipase A2 here in green. Okay, right, so basically the MAP kinase enzyme, once it's been activated within these mast cells, is then going to phosphorylate, because after all it is actually also a kinase enzyme. Specifically, MAP kinase is just a serine-threonine kinase, so it isn't as fancy as MEK. Okay, it's going to phosphorylate serine and threonine residues within phospholipase A2, and again, I don't know the exact number of serine and threonine residues that actually phosphorylates, but the principle is what's important, which is that MAP kinase phosphorylates the phospholipase A2 enzyme, and this activates the phospholipase A2 enzyme, which is now going to work on its substrate, which is within the phospholipid bilayer. Okay, now, when we were discussing PIP2, I said that PIP2 was quite a rare component of the phospholipid bilayer. It was a component, it was a normal component of the phospholipid bilayer, but it was a rare phospholipid in the phospholipid bilayer. The phospholipid that phospholipase A2 is going to work on is the most common phospholipid in the phospholipid bilayer. Okay, and it's what is known as phosphatidylcholine. Okay, and phosphatidylcholine for short is usually just abbreviated down to PC. Okay, so P for phosphatidyl and then C for choline. And because this phospholipid is so important, and we'll continue this discussion in the next video.